All right, so I know that it is just the first day into spring break. I don't anticipate anybody's going to show up for this review. That's okay. I'm still going to report it and post it for you guys. So I'm going to go ahead and get started. Week 9, we're going to be getting into a review for the content for week 9. In week 9, we covered chapters 12 and part of 15. In week 8, we covered chapter 11, and we didn't give a review for that one due to circumstances or for things that I had to take care of on that Friday. I wasn't able to fit it in. So we're just going to do chapter 11 and 12 for this review, since we haven't yet gotten through chapter 15 in its entirety in any of my traditional sections. We're going to wait and put that one off for review for next week probably. And I usually like to give a big overview of how all of the immune system works all together. And we'll probably do that whenever we get back from spring break and we have done at least chapter 15 and at least part of chapter 16. So we'll worry about that whenever we get to that. But so for now, we're going to review chapters 11 and 12. All right, I'll start off with chapter 11, so stay 11 there. And this is the chapter on microbial control. Our major topics for this one, we had our terms for what was involved in microbial control. And our terms included like asepsis, which, why is this a highlighter? Sorry, I'm a pen, not a highlighter. Asepsis, which is referring to removing microbes from living surfaces like living tissue, like skin. Disinfection is the same thing, removing most of the microbes from an inanimate surface. So if it's a skin living surface, it's asepsis. If it's an inanimate surface, then it is disinfection. Neither of these achieve sterilization. And sterilization is the complete and total removal of all living microorganisms, including those that are dormant, the endospores. The endospores are notoriously difficult to eliminate. And when you are sterilizing something, your goal is essentially to remove endospores. And if you are able to remove endospores, you're removing everything else. So I should probably point out to you when we talk about some of these methods that do not sterilize, like pasteurization, when we pasteurize our milk. And then we've already talked about one of those zoonotic diseases that's carried within livestock. That would include anthrax. So anthrax is caused by an organism called Bacillus anthracis. And it can cause all sorts of levels of anthrax infection. And the way that people tend to get infected with anthrax is going to actually be through exposure to livestock. And so the livestock are walking around in the land and they're coming into contact with the endospores of the Bacillus anthracis bacteria. Those endospores are hardy, they're going to hide out. They're just waiting to get inside of maybe an open wound or something so they can take advantage of this new living situation. And so essentially what happens after that is the cattle can become infected and then the person can become infected as a result. It's typically a zoonotic disease. And then people have figured out that if you can dry out those endospores, then you can disseminate them in mail and things like that. And it became a pretty severe bioterrorist threat. Anthrax is a pretty serious disease, but it is an endospore disease as well. So that just means it's difficult to get rid of those endospores. They're all over in the environment, and especially in livestock areas. And so the problem is that our milk comes from livestock that are walking around in these areas that have these endospores, potentially just resting in the land where these livestock are growing up. And so pasteurization is meant to get rid of pathogens, but it doesn't necessarily get rid of all of them. It doesn't sterilize. And so which organisms do you think aren't getting sterilized out? It's going to be endospores. So there's a risk there. And so it's, of course, the responsibility of the farmer to have their products tested through food testing to make sure that we aren't contaminated with these things that could be potentially seriously pathogenic and stuff like that. So if you are somebody that owns dairy cattle, I would not drink raw milk. And if you're somebody that has been hearing all the benefits of raw milk, I would never drink raw milk. I'm just saying, I, as a microbiologist, would never drink raw milk. I don't have to have everything sterile, for sure, but I'm just saying raw milk is one of those things that will never be on my list. All right, so let's get into the concepts or what we covered in this chapter. Microbistatic and microbicidal, these static and cidal, it's the same meaning as what we had in the lab. If something kills a microbe, it is cidal. If something prevents it from growing, it is static. And if you can use static cleaning agents, cleaning methods, and antibiotics, any of these drugs that we talk about in chapter 12, you can use these things if they're static. And if you use them long enough, eventually cells don't live forever. Endospores, they can hide out for a very, very long time. Those endospores can hunker down and outweight any of our cleaning methods. So static isn't going to work on an endospore, but most microbes that can't go into endospore mode, using static methods for long enough periods of time can lead to elimination of infection and things like that. All right, so when you're using heat, so our physical methods that we talked about, our physical methods, those are going to be heat and radiation, and filtration is another way to physically separate the microbes from there. Filtration won't kill the microbes, but it does separate. So that's a process that we call decontamination, when you're separating out the microbes mechanically. Then we have then we have like using salt to pack things. So that is using osmotic pressure to cause the cells to shrink. So that is actually a physical method of preservation. Cold is a method of preservation. Desiccation, drying something out, is a method of preservation. These things will be static, right? Cold and desiccation, things like that. They stop the growth, but they don't necessarily kill very many microbes, at least not very well. And whenever we're trying to kill the microbes, again, if our goal is sterilization, you're going to have to kill all of the microbes, including the endospores. One of the most effective ways of controlling microbes is by using heat. And so when you're using heat to control your microbes, these bring into our concepts of our thermal death time and our thermal death point. And so your thermal death time is the amount of time that you have to treat your sample to get rid of all of this specific microbe. So it's usually per microbe. Each microbe has thermal death time associated with each temperature that you're looking at, and a thermal death point. So a death point is the temperature for that microbe, the temperature that you have to achieve to kill all of that microbe in 10 minutes. So the thermal death time is how much time does it take to kill all of this species of microbe. How much time does it take to kill all of the species of microbe at this specified temperature? The thermal death point is a different thing. This is at what temperature can I kill all of this microbe if I'm only given 10 minutes to do it? And so they're different things. A lot of heat and how it actually is going to work with our disinfection and sterilization process. Heat works by typically denaturing proteins. So the heat with those weak hydrogen bonds that are holding together the major aspects of our protein structure, we can weaken those bonds even further by adding in heat. And so that's the physical way that we can cause denaturation of the protein. So changing its shape, by changing its shape, then changing its function as a result. So it's the goal a lot of times with our heat. But other physical methods using radiation in the cold and the desiccation and the things like that. I feel like heat is pretty straightforward as far as then you add in heat, it disrupts your hydrogen bonds in your proteins. And when you add moisture to that heat, now you're interfering with the hydrogen bonds of your protein by adding in, how water can interfere with that. And so water helps with coagulation of the proteins by adding the heat. So heat is significantly more effective at causing denaturation of proteins if you add moisture in. Very, very significantly more effective. So if we're talking about 121 degrees Celsius, at a higher pressure, typically is when we'll do it, higher pressure in order to achieve that temperature. At 121 degrees Celsius, if you're adding moisture in, if you're adding water in, then you would be able to sterilize something within 15 minutes. If you're doing that same temperature, 121 degrees Celsius, and you're treating something with dry heat, no added moisture, it's going to take 600 minutes. So it's 10 hours. So significant difference there. So adding in moisture makes things significantly better at controlling microbes. And but not everything that you're trying to sterilize can manage being exposed to the moisture. So when you've got metal instruments that you want to be able to sterilize repeatedly, eventually that moisture is going to cause rust, and that will cause the degradation of the material, and that can cause other problems with it as well. So not everything can be treated with moist heat or pressure. Not everything can withstand the pressure needed in order to get that temperature with moisture. So those are all things that we would have to consider. The most commonly used moist heat method for sterilizing is the autoclave. So it combines pressure, heat, and moisture in order to achieve that sterilization, typically within 15 to 20 minutes at 121 degrees Celsius, 15 psi, which is an elevated pressure that helps achieve that. Incineration is a method of literally using high levels of dry heat in order to oxidize and then effectively burn organic matter to ash. So we kind of know how that works. We incinerate whatever's on our loops in lab, and that will sterilize. Boiling is a moist heat method that does not sterilize. Boiling does not sterilize. Boiling does not sterilize. It does not sterilize. It doesn't sterilize. I don't care if you've been brought up by mama and she's sterilizes the baby bottles by boiling them. Boiling does not sterilize. It sanitizes or disinfects. That's the same thing. So that's what that will do. It doesn't get rid of endospores. So if you were boiling something for probably, I don't know, if you were going to boil something at boiling hot temperatures for like a year, you could probably sterilize it, but within practical use sense and how people will be using boiling for 30 minutes, an hour, or something like that, it's typically not used in a way that can achieve sterilization. There is a way to make it work to sterilize, but then it's not true just boiling. You add on other steps. Like if you boil something and you wait a certain amount of time, and then you boil it again, and you wait a certain amount of time, and then you boil it again, 30 minutes each time, and you're waiting this amount of time in between, then you can catch the endospores of certain bacteria. And so whichever endospores you're targeting, you'll have to target the window whenever they're turning from endospores into vegetative. So you can boil away the vegetative cells. And you're going to do it multiple times. And that's a process called tyndallization. It's a separate process. That is not just boiling, but that is using boiling and timing. And you can sterilize with that. Tyndallization is what that's called. But people normally aren't doing this. And it requires a pretty thoughtful knowledge of the endospores that you're trying to treat, right? So knowing their life cycles and things like that. So for all intents and purposes, do not rely on just boiling for sterilization. It doesn't sterilize. And for my purposes on my test, guaranteed that if I talk about boiling, that the answer is it doesn't sterilize. Okay. So that's a moist heat method that doesn't sterilize. Pasteurization, which we mentioned just a minute ago, is a moist heat method where we're cycling temperatures, just like I described in that tyndallization situation. Except we're not getting that high. We're not going to boiling. And so we're cycling from lower temperatures, let's say 60 to 70 degrees Celsius, so below boiling, and cycling that to 30 seconds to maybe 30 minutes tops, depending on what temperature you're at and what type of pasteurization procedure you're using. But you'll go to that sub-boiling temperature for a short period of time, cool down, and then repeat this multiple times. And it will eliminate most of the bacteria, but it doesn't sterilize. But this doesn't disrupt the proteins that are typically found in that liquid. So the reason that we pasteurize milk and don't sterilize it is because there's proteins in your milk that will get destroyed by our typical sterilization procedures. So we don't sterilize milk because, number one, it affects the proteins in the milk if we do that. Number two, the microbiome situation that comes along with having some microbes in our food and drink is actually beneficial. We don't want to sterilize everything. Otherwise, you're not building up a healthy microbiome or maintaining a healthy microbiome in your gut. So the microbes can be there for beneficial reasons, but trust me when I tell you, the more likely if you are introducing a situation where your microbiome related gut bacteria can survive, you're also creating a situation where the pathogens can survive. So you have to find a way to sort of balance that. And that's kind of what pasteurization achieves. All right, moving on, we're going to talk about our radiation. There's two types of radiation. There is ionizing and non-ionizing. Ionizing radiation, these waves, which will be the gamma rays and the X-rays, what they do is those energy waves knock electrons off of the molecules in your body, and that creates ions. So an ion is a molecule that has lost or gained an electron. And we could define it further, right? If you gained the electron, that is, again, that's gained. I have to still talk it out to myself, y'all. Look at that. If you gained an electron, you're reduced. Leo says If you lost the electron, then you were oxidized. And so ionization involves that process. So if we have energy waves shot into your body, kicking electrons off of just random molecules in your body, creating ions off of these random molecules, it could even be freaking carbon molecules, causing massive damage to these molecules in your body by kicking these off and causing these ions, because these ions are not stable. They didn't want to be ions. If they wanted to be ions, they would have been ions. And so we're forcing them to become ions, and this causes damage to molecules nearby, and it can wreak havoc and tear apart, literally tear apart your DNA and your proteins and your cell structures. And this damage can be so severe that your body literally cannot recover from it. And so you'll see development of cancers or very, very quick deaths at high enough levels of ionizing radiation. And the way that these waves work, these energy waves, if they get to a certain level, basically, of ionization, so shorter wavelengths of energy, the shorter the wavelength, the more damage it causes, and also the less likely you can block it with like lead. So X-rays can be blocked with lead, but other stronger types of ionizing radiation can't be blocked by anything that we know of in all of the world, universe, everything. So, but that means that if we can control the radiation, we can focus it onto certain things, and that radiation will penetrate that item. And now you can sterilize literally every single aspect of that item without having to subject it to heat. So we call this cold sterilization. So ionizing radiation is great for cold sterilization, and you can penetrate with it really well. You just don't want to do it to a living organism. And when we have, unless you're trying to kill it, right, which is the point with the microbes. Then we have non-ionizing radiations. This includes UV radiation, like from the sunlight, or when we have UV lamps. And so UV radiation works a little differently. It still excites molecules. It just doesn't cause that level of damage. It's not causing the molecule to kick off electrons or something. It's just exciting molecules that are right next to each other, especially those thymines, the T's in your DNA. So those T's get excited from UV radiation, and they cause covalent bonds to form between the T next to each other instead of the A, hydrogen bonding with the A across from them. And this can cause pretty significant damage if it isn't controlled. Your body has mechanisms in place to fix that sort of damage, but if it doesn't get it at the right checkpoint or it gets missed somehow, then this can cause mutations in the DNA because we're not having proper T to A, binding across the way. And so that can lead to mutations in the DNA that can lead to cancer. UV radiation doesn't penetrate clear glass and things like that if the UV rays can get through that. But as far as most like solid substances, UV radiation doesn't transfer through most solid substances. So it's great if you can expose a surface for an extended period of time, 30 minutes, an hour or more, to UV light, then you can achieve sterilization of surfaces, but it doesn't penetrate that way. Not like ionizing does. All right, and so so I skipped over our dry heat. I mentioned how dry heat works, but I do want to mention that dry heat can be used to sterilize if you're doing it for long enough. So again, because you have to go longer, because you don't have the moisture in there, that you're gonna have to treat something with dry heat for a much longer time. And if your substance can withstand that. So if glass, metal, things like that, you can put it in an oven for 4 hours and it's gonna be fine. But certain plastics aren't gonna survive that, right? So it has its limits. Ionizing radiation, it's not cheap necessarily to always just do that. But if you have items that might be consumable that you need to be sterile when you get them out of the packaging, you can take this, like, whatever this is, hand strengthening toy. Take this hand strengthening toy that I use to fidget with, and then wrap it in some your plastic or paper on one side, seal it in some plastic, and then put it under the ionizing radiation. And everything about this will become sterile. The inside of the package that it's in will be sterile. The item in there will be sterile, and in the middle of it will even be sterile if it wasn't previously, because the ionizing radiation can penetrate that whole thing. So we use this very commonly for things like needles. When you get a needle out of one of those like paper and plastic packages, it's been irradiated to sterilize it. We did not use heat on it. We used radiation. Probably a considerable amount of items that you use on a day-to-day basis, you don't even realize, have probably been irradiated before you came into contact with them. It's really easy to sterilize that way. But if you are in a hospital where you're reusing items repeatedly, irradiating something might not be as practical. So then an autoclave is a little bit more practical because it only takes 20 minutes and you can control that and maintain that and then reuse things repeatedly that way. Eventually, if you irradiate something repeatedly, you get damage from irradiation even on this inanimate object. It's still made of molecules. We're still causing ions while shooting it at it with our ionizing radiation. So eventually this can lead to degradation of material if you're having repeated irradiation. That's kind of why we typically only use irradiation on our consumables, because really just repeatedly irradiating something, even when it's inanimate, even when it's just an object, that can cause damage from those electrons being disturbed. All right, let me see, get back on track. Filtration, we're mechanically separating, like literally by a filter, trapping microbes on the filter and letting the liquid pass through. You're literally just separating the microbes from the liquid. If your filter pores are small enough, you can trap anything and separate it out of that liquid. So you can achieve sterilization if your filter pores are small enough that any virus could not possibly pass through it. You could potentially filter liquids and sterilize them that way. When you get to that size of a tiny little pore, air doesn't flow effectively through that very well. So you can't sterilize air because we just can't the pores are too small to let the air pass through freely to effectively achieve that. But you can decontaminate the air actually pretty effectively using HEPA filters. They don't sterilize, but they come pretty close. But in order to maintain that airflow, the pores just can't get small enough to eliminate everything. All right, let's move on to our chemical methods of microbial control. Our major groups, we have them out here, but I'll write them out to the side just so we can refer to them really quick. The halogens, these are going to include chlorine, bromine, fluorine, and iodine. They're literal like halogens on the periodic table of elements. Remember how our periodic table of elements are separated out basically by the common groups. And so that one of those groups is the halogens. You also have like your heavy metals and things like that. But the halogens are going to include chlorine, bromine, fluorine, and iodine. So those are the actual elements. And so we can have those incorporated as other chemicals, but they're still acting based on the fact that they are halogens. These guys at the high enough concentrations, and if you're exposing something for a long enough period of time, can sterilize if used appropriately. Most of the time we don't use them like that, but we usually water down our bleach. You're probably not going to sterilize something with typical watered down household bleach, but you will disinfect it very effectively. And if you let it sit long enough and you maintain that concentration of that bleach on there, if you let it sit long enough, eventually it can sterilize. Because these guys do have sporicidal activity. Sporicidal, cidal being killing, spore being endospores. All right, so that's those guys. Then the phenol-based compounds. Phenol, it's like, all right, so our phenol and our phenolics. So phenol is like one of these guys. You get a benzene ring. You've seen this in chemistry, don't you love it? You've got like alternating double bonds. All right, so with phenol, you have an alcohol group off the sides instead of an OH group. That's phenol. And due to its like basic like chemical nature, this can be involved in denaturing proteins and disrupting like membranes and things like that. This does not sterilize. So I'll put check mark next to halogens. These guys can sterilize. Phenolics, so those are phenol and molecules that are derived from phenol called phenolics. Those guys do not sterilize. So I'll put an X next to them. No, they're not going to achieve sterilization. They're very good for disinfection, but they're also pretty toxic. So we don't see a whole lot of phenol anymore. And even our phenolics, we keep them limited to particular uses. One very commonly used phenolic would be chlorhexidine. You find it in Hibiclens. And I think Peridex is one of them. It's used as like mouthwash and stuff like that. So chlorhexidine is pretty commonly used. If it's at too high of a concentration or if it's used too much, then we can see toxicity developing, and we can also have allergies to them. But so those are used often in like skin, like scrubbing. Or if you have a skin infection, you can take a shower and with a body wash that has chlorhexidine in it and that sort of stuff. It won't sterilize, but it's very good at disinfecting, and it's something that you can use on the skin at low enough concentrations. So we did the halogens, we did the phenolics. So next we'll move on to our alcohols. Alcohols don't sterilize either, but they are pretty broad spectrum disinfectants. So they can affect microbes by affecting their cell walls and their cell membranes. And if they're at high enough concentration. And if you have the right amount of water in there too, you can coagulate proteins. So alcohols are most effective when you have 70% alcohol and 30% water. You don't want to go to the store and say, oh, 90% alcohol, that's got to be better. It is not. 70% works the best because the 30% water helps with coagulating the proteins. So you get more action going on than just affecting cell membranes and cell walls, but you need the water there. Just like the moist heat is more effective than the dry heat, the moist alcohol is more effective than the dry alcohol, if that makes sense. So, so those are alcohols. This includes isopropyl and ethyl alcohol. Isopropyl is more effective and cheaper. However, it also dries out the skin more. So if you are using things like hand sanitizer very, very regularly, then this can cause your skin barrier, which if you're a lady and you follow up on skincare, then you know how important your skin barrier is, maintaining the healthiness of your skin to keep microbes from getting in, as well as just keeping the integrity of your skin healthy. So once your skin starts drying out too much, you can see cracks and micro fissures where microbes could get in, increasing your risk to infection, which is the opposite of what hand sanitizers are trying to do for you. So ethanol is better about maintaining your skin barrier, but ethanol is it's more expensive and it's not as effective. So if you're not using hand sanitizer very frequently, then isopropyl is the way to go. Or if you're using it as like skin prep for an IV or something like that, then isopropyl is better. All right, the oxidizing agents. So I'm going to give the example for this one because the obvious big one that we need to know for this one is hydrogen peroxide, H2O2. Hydrogen peroxide. We have a hydrogen per oxygen, hydrogen peroxide. All right, so that's H2O2. These guys literally act as oxidizers. So an oxidizer causes something to lose an electron. So essentially, these guys operate by causing free radicals based on oxygen, and then that causes damage to cells from that. If you're an organism that deals with making your energy from using oxygen as your final electron acceptor in your electron transport chain, I'm just telling you guys, this stuff doesn't go away. Unit 1 stuff, we talk about this stuff all the time. Unit 2 stuff, a lot of that stuff's going to come up all the time as well. So we never stop using those other units. I feel like it's unfair to say that a Unit 3 exam is not comprehensive because it's literally going to rely on you remembering the other concepts. We never stop using it. So Unit 2, we did chapter 10, we learned about our metabolism. If you're an aerobic organism like humans are, then you rely on making your ATP through aerobic respiration, and your final electron acceptor in that process is oxygen. And so you've got to be able to deal with oxygen products that are just made from exchanging electrons and taking and removing electrons associated with oxygen, because it can lead to these free radical intermediates. So we have all sorts of mechanisms in place to deal with that. Superoxide dismutase, catalase, enzymes like this are involved in dealing with that. Anaerobic bacteria, the ones that can't survive in the presence of oxygen, they don't use oxygen, and they can't even manage dealing with free radicals from oxygen. Those are called obligate anaerobes. Obligate anaerobes, they can't deal with oxygen. And so using oxidizing compounds like hydrogen peroxide, these generate oxygen-based free radicals that cause a lot of damage to anaerobic bacteria. That's the idea of how those things work. These work very well against anaerobes. If you use these at too high of a concentration, though, it'll cause damage to your own cells. These at high concentrations can sterilize. If you're buying hydrogen peroxide over the counter at CVS or whatever, that's at 3%. It has to be at least 35%. You can't buy it over the counter like that, just FYI. But it could potentially sterilize. All right, then we have our surfactants or our detergents. Abbreviate these, surfactants and detergents. These can't sterilize, and in fact, they're pretty limited in how they can be used, but they mimic your phospholipids in how they are designed. They have a charged head and a long hydrophobic tail, and so they squeeze in between your phospholipids. These guys actually typically have a positive head, and that actually makes them more likely to be able to squeeze in between your membrane lipids. So that charged positive head is usually based on ammonia compounds, and so we call these guys quaternary ammonia compounds, and they're cationic because they're going to have a positive charge in these environments. So these cationic quaternary ammonia compounds have that positively charged ammonia-based head and a long hydrophobic tail. They squeeze in between your phospholipids and disrupt the integrity of the cell membrane. So now the cell can't control what goes in the cell, and it can't keep what's in the cell from leaking out. So this can cause problems. So surfactants and detergents are often used to weaken the membrane so that other drugs can do their job by getting in or out. All right, the next group is our aldehydes. There's 3 major aldehydes that we had to know: glutaraldehyde and formaldehyde. These guys could sterilize if they're used in the right circumstances. This is definitely a check mark here. The other one, orthophthalealdehyde, OPA, this one is safer to use. Glutaraldehyde and formaldehyde are very toxic. You often have to wear masks if you're using them like actively to prevent the fumes from causing damage to your own proteins and things like this. But orthophthalealdehyde is easier to work. It's way less toxic, but it doesn't sterilize. So that's a downside with that one. All right, our gases, the major gas that we talked about was ethylene oxide. Ethylene oxide. This can sterilize. It's just a gas. You can fill a room with this gas, and then everything in that room gets sterilized. It's really effective. It's also explosive and carcinogenic. So we only use these in very limited situations. We often use this gas to sterilize BSL-4 labs. So in labs where you have to work with it with that full positive pressure suit on and everything like that, like what we see in like Outbreak or whatever, that in those sorts of labs, if you need to clean that lab, it's way easier just to fill that lab with this toxic gas than to try to clean in all the nooks and crannies. We'll just saturate that whole room with this gas, let it sit for a long, long time, hours typically, and then we can vent that gas out and even treat it and make it safer for the environment. But it has to be used in a very controlled way. And it's a real big issue with safety. And then our last group is our pH control group. So these are acids and our alkalis. Typically with how you're usually using these are not going to sterilize. They'll just disinfect. This is just going to control, like you're in a solution. how many hydrogen ions are available to interact with your proteins. If your body is typically at 7.35 pH, a pH of 7.35, deviating from 7.35 to 7.45, deviating from that range can be devastating to your proteins. And so introducing acids and alkalis can shift the pH for things like bacteria or even endospores or whatever. So I want to bring something up very, very quickly. You might not be able to kill endospores necessarily by just introducing them to a weak acid, but if you can keep the pH low enough, you can prevent them from being able to turn into vegetative. And the vegetative ones are the ones that make the toxins. So what's the risk? So then that's the situation when you are a brewer, if you're making a product that has a low pH, if your pH is below 5, Clostridium botulinum can't grow. I'm not guaranteeing you that the Clostridium botulinum endospores aren't there. They might be, but they can't turn into the vegetative form, which is what makes the toxin. So if you can prevent them from growing and keep your liquid and your pH below 5, which it often can be whenever you're fermenting something because you make acid as a byproduct, then that controls your risk for botulism. I'm moving on. Chapter 12, quick review of these guys. All right, so we had our antibiotics. Antibiotics are only going to be used for bacteria. Our antibiotics fell into the following categories. You either affected the cell wall, the cell membrane. Oh, come on, pen. I forget that it does this. I just got the hankering to go get sandwiches at Lee's. Cell wall, the cell membrane. It could affect your protein production via controlling your ribosomes. It could affect DNA or RNA synthesis, so nucleic acid synthesis, or it could affect your folic acid synthesis pathways. That's all the ways that antibiotics work. So these antibiotics, these only treat bacteria. Then we had our groups that were our antifungals. How do I establish this? Whatever, antifungal. Then we had our groups that were antiviral. And we had groups that are antiprotozoa. And we had groups that were antihelminths. Once you start getting into these categories below the bacteria, you got problems because our cells are eukaryotic cells. Fungi are eukaryotes, protozoa are eukaryotes, and helminths are eukaryotes. Helminths are way more closely related to us than any of these other guys because helminths are animals. So the closer you get to the helminths on that, the more complicated the organism, the more likely that drugs that you use to kill those guys could be toxic to you. So that creates a unique problem. Our cells are very dissimilar to our eukaryotes, our prokaryotes, the bacteria and archaea. So they provide a little bit more unique development of drugs to attack them, which is why we, kind of have such this wide group of them. This idea that you want to kill the pathogen but not be toxic to your patient is called selective toxicity. The ratio of when your drug becomes toxic to people versus where your drug is actually effective against the pathogen or whatever you're trying to treat. The concentration of drug to treat what you're treating versus the concentration where it will become toxic to people. That actual ratio of those numbers is called the therapeutic index. And the difference between those, let's say it's 0.25 to kill the microbe, and at 1, which is higher than 0.25, at 1 milligram, it is toxic to people. Well, you've got everywhere from 0.25 up to 1 milligram to work with. That's called the therapeutic window. So these are all concepts that work together, but they are different. You need to know the difference for the exam, just so you know, start prepping for Unit 3. All right, so let's start cranking through these. All right, and we know about the Kirby-Bauer assay. If I need to start talking about the Kirby-Bauer assay again. We know you create a lawn of bacteria with your patient's sample. We stamp it with some antibiotics. And where the bacteria doesn't grow because the antibiotic is preventing it from growing, that's the zone of inhibition. We talked about how you would determine if your antibiotic is killing the organism or just making it not be able to grow. And that is cidal versus static. And so we have these concepts that we can build off of to guide us in how we're going to choose our medications. We have to be sure that the medication isn't going to be too toxic for the patient. That's being considerate of that selective toxicity. In order to be able to do that, then you can figure out your therapeutic index and then work within your therapeutic window. Superinfection happens when you use one of these antimicrobial drugs and you kill off your natural biota, and then what's left can overgrow. This is common in yeast infections. You take an antibiotic, you kill out the bacteria in your flora, and now the yeast that's left can overgrow because yeast is fungi. So that's a great example of that. If you're an organism that generates an antibiotic naturally as just part of your life, then you have to be resistant to that antibiotic that you make. So if you're creating an antibiotic that inhibits ribosomes, you better have something in place in your own ribosomes that will inhibit that from working on you, right? So that's called intrinsic resistance, all right? And then you can also have acquired resistance. So this means you picked up genes from other microbes that you come into contact with. Either they're giving you a plasmid, so you could have exchange of plasmid through the sex pilus via conjugation, literally shuttling it through that channel between the cells. You could have picking up DNA from outside the cell, that's transformation, or you could have gotten your genes from transfer of genes through the phage transmission process. That's called transduction, and remember, can lead to lysogenic conversion of bacteria that way. All right, so now let's jump into our examples for these guys and what you need to know for these guys. If it is on your study list, you need to know what, if it is antibiotic or an antifungal or an antiviral, whatever, you need to know what category it falls within here. First, what organism does it affect? Bacteria, fungi, protozoa, viruses, or helminths. Then, if it is an antibiotic that affects bacteria, you need to know if it affects the cell membrane, the cell wall, the ribosome protein synthesis, or nucleic acid synthesis, and folic acid synthesis. All right. So, let's give our examples for these. Our cell wall antibiotics. The antibiotics we need to know for the cell wall are psyllins. Those are all derived from penicillin. If it's named psyllin, all of the psyllins affect cell wall synthesis, period. And there are any of them that aren't penicillin were derived from penicillin, okay? And these are going to have a special ring structure in them called a beta-lactam ring. There's other drugs that also have beta-lactam rings, like the cephalosporins and the carbapenems. These also affect the cell wall. Two other drugs that I would be aware of that affect the cell wall are bacitracin and vancomycin. For the ones that affect the cell membrane, the one you need to know is polymyxins. It's just one. For the ones that affect the protein production by affecting the ribosome's ability to make proteins, these are aminoglycosides, cyclins like tetracycline and glycylcycline, and then mycins. So the mycins, almost all of them, most of the mycins will affect the ribosome function, except for vancomycin. Vancomycin affects the cell wall. That's the only deviant there. All right, our nucleic acid synthesis inhibitors fall into a group known as the fluoroquinolones. The fluoroquinolones include any of the drugs called fluoxetine. So if it has fluoxetine in the name, it is a fluoroquinolone, and the fluoroquinolones inhibit nucleic acid synthesis. Then we have our folic acid synthesis pathway inhibitors. So these are competitive inhibitors. They bind at the active site on the enzymes involved in how bacteria make folic acid. We as human beings do not make folic acid. It's provided to us in our diet. And so these drugs target a pathway that's unique to bacteria. And so these are your sulfas and trimethoprim. If you combine trimethoprim with sulfamethoxazole, which is sulfa, then you are targeting blocking the folic acid synthesis pathway at two different points with two different drugs. They work a lot better together when they do that. When you combine those two together, trimethoprim and sulfamethoxazole, those guys create Bactrim. That's the trade name for it. I'm pretty impressed with how this, translator, whatever, however you want to call it, CC is working down at the bottom of the screen. It's actually picking up on these words, man. All right, so anyways, so those are all of our antibiotics. Then you need to know what the antifungals are. The azoles, if it has azole in its name, there's only one outlier. So the mycins, the one outlier was vancomycin. It affects the cell wall. So you got to know that one separately. The azoles, the one outlier is metronidazole. Metronidazole graduated and moved to the big metro, and it treats protozoa and anaerobic bacteria. So it's special. So just metronidazole. So our deviant for our azoles is metronidazole, which is right below where I'm writing this. And it treats amoebas and anaerobic bacteria. All right. But other than that, your antifungals are the other azoles and amphotericin B. That's all you need to know for the antifungals. You don't need to know how they work. For antivirals, the antivirals that you need to know are Tamiflu or Relenza. These block neuraminidase action in flu. Neuraminidase is an enzyme on the flu virus that breaks down mucus and helps with entering and budding out of the cell in the viral life process. Acyclovir, it inhibits nucleic acid synthesis in herpes viruses and other DNA viruses. This includes valcyclovir, which is Valtrex. If it has vir in the name of the drug, it's an antiviral, by the way. And then we have drugs that you don't need to know the individual names of, but you need to know that we have special drugs that target HIV that inhibit the reverse transcriptase, which turns its mRNA into DNA. It inhibits the integrase or integrase. That's an enzyme that inserts that new DNA into the genome. Now, reverse transcriptase and integrase are enzymes that do not exist in our cells naturally. Unless you have HIV, you cannot turn mRNA into DNA, and you couldn't put that DNA into your body. Your body doesn't have that ability to do that. Viruses have to have special enzymes that allow them to do that stuff. It doesn't exist naturally in your body. And it's not like if you have these infections, that these things are being made all the time and your body could just use it for whatever either. So as it turns out, if you have mRNA introduced into your body, your ribosomes might be able to read the mRNA and make the product from the mRNA, the protein, but it can't turn the mRNA into DNA and then insert it into your DNA. It is literally impossible. It's just as impossible as fairies flying out of my butt right now. It's like not a thing. So you can't get COVID mRNA inserted into your DNA, period. It doesn't, it's impossible. FYI. I'm glad that we covered that. All right, so moving on. So then we have, that's fine. So we'll target those special proteins in HIV with our HIV antivirals. And that pretty much covers the antiviral stuff we have to know. And you're welcome for that, because let me tell you, there's so many different antivirals and they're usually very specific to viruses. Like this antiviral works with HIV. This antiviral works with flu. They're usually not very broad spectrum. Not like a lot of our antibiotics are. All right, and then our antiprotozoas, we already talked about metronidazole. It affects amoebas, which are a type of protozoa. It also, antiprotozoal that you need to know about are the quinine-based antiprotozoals, and these treat malaria. So malaria is a protozoa, and it affects your blood and your liver. It's a pretty serious disease. It can be very deadly. It's pretty chronic. It's pretty difficult to treat. But the drugs that we use are based on a compound called quinine. And so those drugs that are derived from quinine are going to have quine in the end of the name. So chloroquine or primaquine. So there's those. And then we have the anti-helminth drugs, and the ones that we would need to know for that would be like praziquantel and ivermectin, very commonly used to treat worm infections in people and in animals. Okay, so we're going to move on now to our missed topics. So for chapter 11, knowing what things sterilize and what things disinfect, remember this was in our chapter 11 slides. This is a great chart to refer back to find out if you think that something could potentially sterilize. So UV radiation, you see in our chart, I want to point something out really quick. UV radiation, you see how it says disinfection? You're only going to sterilize surfaces, and that's only if you are exposing it to the UV radiation for very extended periods of time. Typical use of UV radiation doesn't sterilize very well. And so we don't typically use it for sterilization. So I just want to be very clear about that. Could it potentially sterilize surfaces? Potentially. But this is long terms of exposure. So this one's kind of like one that's on the fence, the UV one. Anyways, no, it doesn't help. Maybe it makes it more confusing. And I apologize for that, but I'm just trying to clarify for you. All right, but if it sterilizes men, and we commonly use it for sterilization, it'll tell you sterilization. All right. So if we talk about steam under pressure, this is your autoclave, right? Or boiling water, hot water. These things don't sterilize. If you're not adding the pressure, then you're only going to stay at 100 degrees Celsius. In order to get high enough to where you can actually sterilize and kill the endospores, you need to add pressure into there. And then, of course, adding moisture as well. So this is a great chart to come back to be able to delineate what can sterilize and what can't. So we have methods which sterilize and methods which don't. So if you sterilize, incineration, dry oven, autoclave, ionizing radiation, only surfaces for your non-ionizing radiation, which is your UV, ethylene oxide gas, chlorine, our halogens in general, hydrogen peroxide, formaldehyde and glutaraldehyde, and then filtration with small enough pores. These things can sterilize if you are using them appropriately. So when we say hydrogen peroxide, I don't mean, man, guess what? Professor Yost said that you can sterilize with hydrogen peroxide. If your hydrogen peroxide is at 35% or higher, yes. If you're going to CVS and buying 3%, which is what you get over the counter, no. So if you're using them appropriately and for a long amount of time and with high enough concentrations, these things can sterilize. All of these can fail if you're not using them appropriately, all of them. So just want to keep that. But then there are other ones like alcohols, which are never going to sterilize. All right, so I've made this list here, the ones that could sterilize, and the one, if it's not on the sterilized list, which is this top list here, if it's not on this list, it doesn't sterilize no matter what. So let's move on. Which of the following methods is most appropriate for sterilizing? Can ionic radiation sterilize? This is one of those moments where if I was taking the test, I'd have memorized some sort of acronym on all of my sterilizing methods. So this says I dare chaff, okay? Ionizing radiation, dry oven, autoclave, incineration, that's what this was. Incineration, I was really confused as to what was going on there for a second. So this is incineration, dry oven, autoclave, radiation in general, if used appropriately. Surfaces will only be for the non-ionizing, but ionizing can penetrate. I dare, what was the E? Ethylene oxide, and then C was chlorine. So this could be any of your halogens, but chlorine is the best example we have. Chlorine, hydrogen peroxide, the aldehydes, and filtration. I dare chaff, whatever. So that's our list of what can sterilize. Let's write it up here. I dare so we can recall it if we need. Chaff. Right. So which of the following methods is most appropriate for sterilizing? In our list, the R was radiation. So we know that this can sterilize. Ionizing radiation can sterilize. Boiling, was that on our list? We don't have moist heat on the list. We have autoclave on the list, not boiling. So no. Is alcohol on our list? No. Aldehydes are, but not alcohol. Is pasteurizing on the list? Again, we didn't put moist heat. We only put autoclave. So it's only going to be ionizing radiation, which can penetrate. It's pretty effective. In a clinical setting, which sterilization method is commonly used to ensure that surgical instruments are free of all microorganisms, including bacterial and spores? So it has to be one of these guys. And it's going to be something that is commonly used for reusable items. Because you typically don't just get rid of your surgical instruments, you can reuse them. And so what do we use in a hospital environment to ensure that our surgical instruments can be reused by sterilization? So it's not going to be pasteurization because this doesn't sterilize. It's not going to be boiling because that doesn't sterilize. So it's going to be between autoclaving and UV radiation. But the difference here is UV radiation is less reliable than our autoclaving. And so like we said, UV radiation is our kind of gray. Come on, pen. This pen is killing me, dude. Ooh, can't draw a star with it, apparently. I'm doing each line individually. So anyways, UV radiation is only going to get at the surface where it hits. So here's the problem. If you put your surgical instruments and put it into a box and shine UV radiation at it for an hour, the entire underside of your surgical instruments didn't get exposed to the UV radiation. But if you put it into a moist heat chamber with pressure, that anywhere that moist heat touches will get sterilized, including anything underneath it. All of that should be perfectly sterilized inside and out. So we use autoclaving for that. Next question. Using osmotic pressure to control microbes. So this is basically exposing microbes to high concentrations of salt or sugar. And that by putting something in a high salt or sugar environment, it will draw the moisture out of the cell and cause the cell to shrink. And this is a process called plasmolysis. This is useful for preservation. It's not particularly great at killing the microbes, not sterilizing level, right? But it can definitely be useful for preventing them from growing, which might be all you need to do, because if they can't grow, then they can't make their toxins and things like that. Again, not a complete sterilization. It's just going to be a partial sort of a situation, but it is effective enough, and you can preserve the food this way for very long periods of time. The microbes can't grow at all. So salty jam stops microbes is our mnemonic here. So we're going to bring that over here with us. Salty jam stops microbes. This is reminding us that high levels of salt or high levels of sugar will stop our microbes from growing. So our questions are, how does osmotic pressure help in controlling microbial growth in food preservation? So like we said, salty jam, so high salt or high sugar, what it's going to do is draw the moisture out of the cells because the salt in the jam is hypertonic, right? So are we altering temperature? No. Are we increasing salt and sugar concentration, which will cause water to leave the cells? Yeah, we are doing that. It's probably going to be our answer. Let's make sure everything else is taken care of. If you are ever taking one of my tests and you're reading the question and you see one answer and you are like, oh, this looks like it. If you do not read the other choices, that's on you, man. So we want to be sure we're looking at all of the choices. So the next choice, by introducing antibiotics that target bacterial cells. This is not introducing antibiotics. We're just using salt and sugar. So it's not this one. And then exposing microorganisms to UV light. We're not using UV light in this situation. Again, we're just using salt and sugar. So B was our answer. So which of the following statements best describes why high osmotic pressure is effective in preventing microbial growth? So what's actually going on when you're shrinking the cell down by drawing out its moisture? Why is that inhibiting the growth? Is the shrinking So we have high salt or high sugar that's stopping the growth. Is this destroying DNA in that process? No, it's just drawing the moisture out. Are we creating environment with low pH? That shouldn't be involved. We're not changing the pH, we're just drawing the moisture out. Are we creating an environment where osmotic pressure causes the cells to lose water and then dehydrate them and eventually kill them from that? Yeah, yeah. Or are we enhancing the growth of the microbes that are beneficial while inhibiting growths that are harmful? That's not what we're doing. That might be something that we're doing with our probiotics and our prebiotics, though, but not with osmotic pressure. Next, we have hydrogen peroxide. Hydrogen peroxide is one of our chemical methods of microbial control, and it's going to oxidize by creating free radicals of oxygen. And that these free radicals are going to cause damage to the cells around them. So it says bulb valves for endospores. At high concentrations of hydrogen peroxide, it can sterilize. High. We'll write that up here to remind ourselves. H2O2, high concentrations of H2O2 on a pen can sterilize. And it works by oxidizing. And how the oxidation is happening is by creating oxygen-free radicals. And these cause damage to other molecules. That's how it works. All right, so there's our little note that we wrote for the questions. What is the primary mechanism by which hydrogen peroxide acts to control microbial growth? So we're looking for oxidation and oxygen-free radicals here. Does it disrupt microbial cell wall by enzymatic activity? This is asking, is H2O2 an enzyme that destroys the cell wall? That's not what's happening there. It's going to create free radicals that then lead to damage. Is it causing death through production of reactive oxygen species, which are also known as ROSs? These are free radical oxygen molecules. So yeah, it's probably this one. Is it altering pH? We're not affecting pH. We're creating free radicals. Or is it forming a physical barrier? We're not doing a physical barrier. We're causing free radicals. And those free radicals are reactive oxygen species and cause damage to other molecules in the cell. That's how it works, period. So in a healthcare setting, how might we use hydrogen peroxide to ensure that our medical equipment is safe? Do we coat our medical instruments to prevent microbial contamination during storage? Do we coat our medical instruments with hydrogen peroxide? We'll come back to that. Is it applied directly to wounds to speed up healing by reducing microbial load? Now, this should sound familiar because this is probably how you may have treated your wounds as a kid, or your mom did, or something like that. So now we're getting somewhere. Do we use it in hospital settings to disinfect surfaces and sterilize equipment? When is the last time that you disinfected a surface or medical equipment using hydrogen peroxide? It does have these oxidative properties, but you'd have to be using it at high concentrations. This isn't impossible. This is a possibility for this stuff to be using this way. The problem with using it for wounds to speed up healing is that you could possibly be causing damage to tissue through causing injury to your own tissue. And then that could be actually a feeding ground for microbes. So these are issues that we have to consider when we're using this. And as it turns out, this can be corrosive because it's causing damage to the cells as well as other materials and things like that. And so what we will typically do in our medical settings, we can use it for certain wound cleaning situations. We can use it for sterilizing medical equipment, and we can use it to disinfect surfaces, right? We can use it for these things. And so technically, when we're looking at this particular situation where we're choosing, we could go with B, or we could go with C. Are we adding it to solutions to enhance the effectiveness, we typically don't use it this way. And it typically doesn't create a coating on the instruments. It's like a one-time treatment. And then the after effect is you are disinfected or you're not. But it doesn't have a coating that like maintains the cleanliness over time. So B or C are actually accurate in this particular situation. Of course, on the test, you would only have to choose one, but I wanted to walk through some critical thinking with this. All right. And that's not going to be as complicated as what it'll be on the test. Most of the questions for Unit 3 will ask like, could hydrogen peroxide sterilize, you know, at high enough concentrations? And I'll probably be specific about that. If you have high concentrations of hydrogen peroxide at 35% or higher, could that potentially sterilize? I would hope that you would know that it could. That's most of the questions for the Unit 3 stuff for chapters 11, 12, and 18. Chapters 15, 16, and 17 are considerably more complicated than that. They're not flashcard chapters, but these are, right? All right, so therapeutic index. So we know selective toxicity is the idea that you want the drug to be effective against the thing you're treating, but not be toxic to your patient. Therapeutic index is the ratio where it's basically a measure of safety of a drug, where we're looking at the ratio between the effective dose of the drug to treat whatever you're treating. At what level does it become effective? And then the toxic dose, at what level does it start to cause harm to people? So when we look at penicillin has a very high therapeutic index. This means there's a large margin of difference. Like there's a good therapeutic window. There's a big difference between where it's effective and where it becomes toxic to people. Most of the time, the toxic dose of penicillin, we're not talking about allergies, we're just talking about the drug becoming toxic. So most of the time with penicillin, it's extremely high levels of penicillin that you will start to see toxicity. But it doesn't take very much penicillin, very low levels that'll be effective against microbes. That gives you a massive window within which to work. And so that is what we call a high therapeutic index. Is that when those ratios, those numbers are very far apart from one another in that. All right, so what does a high therapeutic index? So when those are seeing that difference there. When your therapeutic index is indicating that there is a level of safety available there, you have a big window to work within. That's what that's talking about. So high therapeutic index, you have a big window to work within. What does that indicate about the drug? Does it tell you that the drug has a high risk of causing severe disease or severe side effects, even at low doses? Does it tell you that the drug has a large margin of safety between the effective dose and the toxic dose? So if it has a high therapeutic index, if you have a big window to work within, what it's really telling you that there is a large margin of safety. There's a large range within which you can work. There's a big difference between where it's toxic and then where it's going to be effective. So low levels of effective dose, and it would take a whole lot for it to be toxic. That's a high therapeutic index. It's a big window to work within. So this is going to be answer B. All right, next question. In a healthcare setting, why is it important to consider the therapeutic index? Because you want to be sure that you're killing the microbes and not causing problems with your patient, right? So does this have anything to do with color and taste preference? Obviously not. Does it ensure that the drug is administered at a dose that maximizes therapeutic effects, the result you want, but minimizes toxicity? This is exactly why it's important. And that concept that this is outlining in B is called selective toxicity. The actual numbers, the therapeutic index, and then the therapeutic window is that range within which you can work. To evaluate the cost effectiveness, we're not talking about cost effectiveness with it, and to predict the drug's effectiveness based on age and gender, this has nothing to do with that. This has everything to do with the difference between where it's effective and where it becomes toxic. So now let's go over our protein targeting drugs. These affect the ribosome function. This includes our aminoglycosides and our tetracyclines and pretty much anything called myosin. This falls into another group, the macrolides. I'm just going to say this, the macrolides are based on like their chemical structure, and we have macrolides that affect your protein synthesis, and we have macrolides that are associated with antifungals. Amphotericin B is actually a macrolide. Amphotericin B is an antifungal. So that's why I don't focus on macrolide side of things, because they have their hands in a lot of stuff. Mysins, aminoglycosides, tetracyclines. That's really the best way to break it down. It says a tiny mouse, but I want it to be like A would be aminoglycosides, tiny would be tetracyclines, and mouse would be mysins. The only mysin that doesn't fall in this category that we care about is vancomycin, and it affects the cell wall. So we have to remember that. All right, so if we're remembering that it is these aminoglycosides, and if I'm talking about aminoglycosides, I will call them aminoglycosides. I'm not going to expect you to know that streptomycin is an aminoglycoside. We're not doing that in my class. It's too much already, right? So I will call it aminoglycoside. If it's a group of drugs called the aminoglycosides, which part of bacteria does it affect? Protein synthesis by inhibiting the ribosomes. Okay, so aminoglycosides are tiny mouse, a tiny mouse. The T was the tetracycline. And the mouse was the mysins, except vancomycin. Okay, so which of these following antibiotics would affect protein production? Now we just got to match it up with one of these things up here. Well, I already found one. If it is not called with these names, does carbapenem, is it called an aminoglycoside? Is it called tetracycline, or does it have mycin in its name? It does not. Neither does ciprofloxacin, and neither does metronidazole. As it turns out, carbapenem affects the cell wall, ciprofloxacin affects nucleic acid synthesis, and metronidazole treats anaerobic bacteria as well as amoebas. So why might tetracycline, we already know it's over here, it's on our list, it's right here. Why might tetracycline be preferred for treating gram-negative infections as opposed to amoxicillin? So if we're referring specifically to gram-negatives, there's probably a reason why we're talking about gram-negatives as opposed to gram-positives. So what's the difference between the gram-negatives and the positives? The gram-positives have a thick cell wall. They need that thick cell wall. If this is an A, I promise. They need that cell wall to be thick. If that cell wall is compromised, there is nothing else to help them out. The gram-negatives, they have very little peptidoglycan cell wall. They have a very thin cell wall. And they also have an outer membrane. So if they lose their cell wall, they've still got that outer membrane to help them out. So they can deal. So that's what we're comparing here. And we've already talked about how the cillins affect the cell wall and that those would be preferred for treating gram-positives. We talked about it in the lab. And so this is the same thing. So why wouldn't you want to use amoxicillin to treat a gram-negative infection? Because they can manage having their cell walls damaged. However, if you use tetracycline, it affects protein synthesis. That doesn't matter if you're gram-negative or positive. You still got to make protein, right? So it's going to work to inhibit the protein synthesis, whereas if we're affecting the cell wall, it won't have as much effect on the gram-negatives. And so now we're going to go through these and make sure we're making sense of this. So would amoxicillin be affected because it's metabolized by the gram-negatives? No. Amoxicillin isn't effective because it affects the cell wall. Gram-negatives, they're not as impacted by that damage. Next choice, tetracycline targets protein production. It does in fact do that. While amoxicillin targets cell walls, it does do that. And gram-negatives have very little cell wall. Exactly. Next choice, does amoxicillin enhance protein production? No, amoxicillin stops the cell wall synthesis. And does tetracycline attack the gram-negative outer membrane? No, tetracycline affects protein synthesis. So B gives us the best answer in this particular situation. Moving on to our nucleic acid synthesis inhibitors. These guys inhibit helicase from unwinding your DNA. So our nucleic acid synthesis inhibitors inhibit both DNA and RNA replication. The larger group is called the fluoroquinolones. And the ones that fall within that group have a name ending in floxacin. All right, so let's move on. It says fluorescent falcons. The fluorescent is for the fluoroquinolones. The falcons is referring to the floxacin side. I don't know if that helps at all, but whatever. So the group is the fluoroquinolones. And again, you just have to recognize them on the list, or are groups that end in the name floxacin. And what these guys do is inhibit nucleic acid synthesis. So let's look through our list. Which of the antibiotics listed below affect nucleic acid synthesis? So we are looking for the group, fluoroquinolones, or individual antibiotics named with the ending floxacin. As it turns out, our first choice is ciprofloxacin. Floxacin, that's our answer. Carbapenem affects cell walls. Amoxicillin affects cell walls. Glycylcycline is a cyclin. It's affecting the ribosome, just like tetracycline. And why might the fluoroquinolones, which affect nucleic acid synthesis, have negative side effects in humans? This is pretty much what the question is asking. Why might fluoroquinolones, which affect nucleic acid synthesis, have negative side effects in the human bodies they're being administered to when their infections are the real targets? Well, if we're affecting nucleic acid synthesis, don't your own cells need to make nucleic acids? In order for your cells to function, your genes code for RNA, right? So, and that RNA is turned into protein. So your genes are in DNA form. The gene is turned into the working copy of the mRNA, and then that mRNA is turned into protein through translation by the ribosome. If you can't make that RNA, you can't make any of your gene products. You can't even make tRNA. You can't even make ribosomal RNA. You can't even make ribosomes. You can't make copies of your cells. You can do nothing. And so we have to be able to make our own DNA, our own RNA. This doesn't just inhibit bacterial cells. The problem is bacterial cells, they are going to be single-celled organisms. And so whenever you inhibit the DNA and RNA processes of a single cell organism, that cell's going to die. We're multicellular, so we can manage losing some cells and then regenerate them, because overall our body is still alive. So we can suffer damage to a point and still survive this, while the bacteria are going to be more susceptible. That's the general idea of how that's going to be functional. But basically, let's come back to the question. So, do fluoroquinolones cause bacteria to secrete toxins? No, fluoroquinolones inhibit the nucleic acid synthesis. Do fluoroquinolones stop protein synthesis by blocking the ribosomes? No. The ones that block your ribosomes are going to be the aminoglycosides, the cyclins, and the mycins, except for vancomycin. The fluoroquinolones, do they affect nucleic acid synthesis? And we also must do that? Yep. Or do fluoroquinolones too quickly get removed from the bloodstream? No, it's the issue with the nucleic acid synthesis that's the problem there. All right, now we are on to our antivirals. Our antivirals are drugs that tend to be unique to specific viruses, and the ones that we have to know are acyclovir, which is typically used for blocking nucleic acid synthesis in viruses. In particular, was developed for herpes viruses. This is the only one that can be used for other virus species that we're talking about in our class. So acyclovir and ones that are derived from it. Tamiflu. Tamiflu has flu in it. What do you think it blocks? The flu. The way Tamiflu works, we have to know for the class. It blocks neuraminidase, and that's an enzyme on the flu virus, which helps with it budding out of the cells. It helps clip off the new budding viruses. If we can't do that, we can't make new viruses. So that's how Tamiflu works. It prohibits neuraminidase from being able to allow new viruses to bud out. And then we have our HIV antivirals. We're not going to talk about the drugs themselves, because this is a continuously evolving field, constantly coming out with new drugs. And we have so many of them. But what almost all of the drugs do will target one of these proteins in HIV. HIV makes reverse transcriptase, which turns its RNA genome It has an RNA genome. It allows it to turn its RNA genome into DNA. Why would it want to do that? Because then it can use integrase to insert that DNA into the human host cell DNA. It can literally insert it into your cells' DNA, into your chromosomes. And integrase is the protein that does that. And then it also makes this other separate thing, protease. And what protease does, when HIV takes its RNA and makes protein out of it, when our ribosomes are reading its RNA and making the protein, I should put it that way, or making HIV protein, it makes it in one big long protein, and then it clips it into the individual proteins. And the thing that clips it is protease. So without protease, it can't clip its proteins and therefore can't make its viral parts. It can't clip reverse transcriptase away from everything else. And so reverse transcriptase can't work. And we can also block the reverse transcriptase itself or block the integrase from inserting the DNA into the cells. These things all work very well together for keeping viral load levels very close to undetectable, but it doesn't guarantee that there wasn't already HIV hiding out in your genome. We have no way of getting that out yet. So that's the problem there. All right, so on with our antiviral questions. Remember that the ones that we talked about were the acyclovir, which we use for DNA, nucleic acid synthesis inhibiting, specifically for herpes, but we have broader uses for it. Then we had Tamiflu, which affects neuraminidase of flu. And then we had our HIV drugs. We're just going to put that there. That could affect integrase, protease, or reverse transcriptase. All right, so which of the following statements is true about acyclovir? So how does acyclovir work? It works by inhibiting nucleic acid synthesis for DNA viruses. So does acyclovir act as a protease inhibitor that prevents protein synthesis? No, it affects nucleic acid synthesis. Is it an antiviral drug that inhibits replication of viral DNA? Yes, it does. That's going to be nucleic acid synthesis, right, for our DNA viruses. Does it bind to the viral envelope? So remember, viral envelope is going to be that membrane that the virus stole from the host. So it's membrane that's outside. Does it bind to that? No, it's interacting with inhibiting DNA synthesis. We're not preventing entry. Or does acyclovir act as a reverse transcriptase inhibitor? We don't have reverse transcriptase. Well, I don't want to get into too much detail with this yet, but this is not blocking viral RNA being turned into DNA. This is blocking DNA synthesis from DNA. DNA replication is being blocked by acyclovir. So we're literally inhibiting DNA replication here. Herpes viruses don't work the same way. We don't go RNA to DNA and then insert our DNA. There's DNA viruses. There's no reverse transcriptase going on there. All right, which of the following choices accurately describes how Tamiflu works? Well, we're blocking neuraminidase since it's going to prevent budding of the new viruses. Prevent budding. Just in case we need it, I'm going to write it over here. This is me brain dumping. This is how I prepare for questions on my tests, especially if I don't just have automatic recall of the thing. So I'll write down everything I know about it. And if it asks a little more detail and I happen to think of another detail, I'll write it down in my big pile of my brain dump. And then I'll use that information to answer my questions. All right, so how does this work against influenza? Does it block the DNA replication? Does that work? That's what's happening. We're not blocking DNA replication with Tamiflu. Does it block viral hemagglutinin? We're not talking about hemagglutinin. When you have flu, you might have H1N1. The H is hemagglutinin, the N is neuraminidase. Tamiflu affects the N, not the H. The H is important for binding to the cells, but that's not what Tamiflu is affecting. This is a drug that inhibits the neuraminidase. Does it prevent the viral genome from integrating into the host genome? Flu doesn't do that. So that's not an issue with flu. And that's not what the Tamiflu does. It inhibits the neuraminidase. Neuraminidase is going to be one of those things that helps with the virus budding out when we're making new viruses. So we're just blocking it from making new viruses. All right, that is it for chapters 11 and 12. Not our longest one, not our shortest one. when we get into talking about chapters 15 and 16, so in our lecture course, we've only made it through part of chapter 15, and we should be able to crank through the rest of it just fine. And we'll definitely have time to get through the rest of all the rest of our chapters. But chapters 15, 16, and 17, make sure that chapters 15 and 16 make sense and how those two chapters work together and everything we're learning about that in order for chapter 17 to make sense. Because chapter 17 is derived entirely from our understanding of what's going on in the innate immune system from chapter 15 and the acquired immune system in chapter 16. It really plays a lot more off of chapter 16. But you cannot understand how the acquired immune system, chapter 16, is going to be controlled unless you understand how the innate system leads into that. And I'm going to go over an entire immune process. And so I am almost certainly going to guarantee that the next review session that we have for chapters 15 and 16 is going to be a very in-depth dive, linking all of our immune knowledge that we're learning from 15 and 16 together. So we'll go from like start of an exposure to something all the way to the finish when you have memory developed. So leading in with all of our functions of our innate response as well as the acquired response. We'll do that. Right now, the only person that's joining me is Jacoby. Jacoby, do you have any questions? All right, awesome. Thank you for joining me, and I will post this recording up for anybody to review too, as well as the AI summary and everything like that. Other than that, please have a wonderful spring break, everybody. Reset, recharge. My biggest recommendation for recharging from having how stressed out as everybody has been lately, find a childhood movie. This is stupid, but I watched this movie Fly Away Home. You don't have to watch Fly Away Home. It's actually better if you pick something that means something to you. And us ladies, I'm not just talking to the ladies, but especially the ladies or people that understand the emotions, have emotional intelligence. If it's a movie that brings happy tears to your eyes, find one of those movies that you have fond memories of that brings happy tears to your eyes and take some time during spring break and watch that movie. It's not just going to be that movie that's going to bring the happy tears to your eyes, but something happens that helps you release the other emotions that we've been pushing down associated with the stress. Just trust me when I tell you that this works fantastically. If you have the ability to put on a bubble bath and watch the movie in the bubble bath, even better, man. Just some snacks, set it up at your bubble bath and watch your movie. Highly recommend. Stupid, but it works fantastically to help dump out all of the emotion that we are taught to push down and not share with our society. So anyways, have a great spring break. And I will see you guys in over a week. All right. Thanks.